Welcome to GI Lecture 2. This lecture is going to be a little bit longer because we're going to cover the stomach, the liver, the gallbladder, and the pancreas in this lecture. So you need to get out your pen and paper and be ready to take notes. Feel free to stop the lecture when you need to, stretch, take a break, and then come back. So here we go. Let's talk about the stomach. So I know in lab that you have looked at all of the anatomy, but I want to review it pretty quickly. So remember that the stomach is where we have mechanical digestion and chemical digestion. Mainly it's the chemical digestion of protein, but there is some lipid digestion that takes place in the stomach. Once the food bolus comes down the esophagus through the cardiac sphincter and goes plop into the stomach is going to churn. Remember that it has muscles in 3 dimensions, and these each contract and relax in their own dimension, and this churns the food. And it takes the food from pieces parts and then turns it into what we call chyme. Notice that our stomach has about 50 milliliters of volume, but it can expand to 4 liters. So if you think about 4 liters is about a gallon, then you can put a gallon of stuff in your stomach. You will be very, very uncomfortable, but you can do it. Here's a great picture. You need to know these parts for the lab exam and for the lecture exam. Please note that there are 4 major parts of the stomach. You have the cardia, the fundus, the body, and the pylorus, or the pyloric area. Notice how the esophagus is going to join the stomach, and that is often called the gastroesophageal sphincter. It's also called the lower esophageal sphincter. It's also called the cardiac sphincter. Just remember that the reason it's called cardia or cardiac is because that's the part of the stomach that is closest to the heart. You'll notice that the stomach has lots of ridges in it, and those are called rugae. And this increases the surface area of the stomach. So the stomach is controlled mainly by the autonomic nervous system, remembering that the sympathetic or fibers are going to inhibit the stomach's physiology, while parasympathetic fibers are going to encourage and stimulate the stomach's nervous system. And the vagus nerve is extremely important to many of our organs, especially to the heart and to the stomach. The blood supply for the tissues come off of the celiac trunk, and the artery that goes to the stomach is called the gastric artery, and the artery that goes to the spleen is called the splenic artery. And then it is drained, the stomach's blood is drained by the hepatic portal system veins. So there are 4 tunics, but what we see is that the muscularis and the mucosa are going to be a little different. The muscularis externa has 3 layers of muscles, 1 for each dimension: circular, longitudinal, and what we call diagonal or oblique. So this helps the stomach with its churning, and also to help the chyme to be moved into the duodenum a little bit at a time. The mucosa layer is modified in that Mainly it has simple columnar epithelium that's composed of mucus cells, and of course mucus cells secrete mucus, plus it's going to secrete an alkaline mucus that has lots of bicarbonate fluid in it, and then it has gastric pits, which look like pores, and at the bottom of these pits are the gastric glands, and the gastric glands have the cells that are responsible for producing all the fluid that we find in the stomach, or the gastric juice. I think it's interesting they call it the gastric juice, and in the pancreas, if it has to do with digestion, they call it the pancreatic juice. Both of these, the gastric and the pancreatic juices, are rich in bicarbonates. So what happens when you add an acid and an alkaline fluid? Think about that from your general chemistry class. This will become very important when we talk about the duodenum and digestion that takes place in the duodenum and the rest of the small intestine. So, what are the kind of glands? Notice that the glands in the fundus in the body of the stomach produce most of the gastric juices. So there are 4 types of cells. The mucous neck glands. Guess what they produce? Yeah, mucus. The parietal cells, the chief cells, and then the enteroendocrine cells. These are going to produce hormones. So here's a lovely picture. See that the gastric pits on the superior side, or the side facing the lumen of the stomach, they're called pits. They look like very large pores. And then you come down and you see the surface epithelium that contains the vast majority of the mucus cells. And then further inferior, we have the mucous neck cells. And the mucous neck cells are where the gastric gland begins. So in the gastric gland, you will have the mucous neck cells, the parietal cells, the chief cells. Notice the chief cells are in purple on this picture, that the parietal cells are blue, and at the very bottom, closest to the capillaries, you'll notice little green cells, and those are called the enteroendocrine cells. So remember, the enteroendocrine is going to produce the hormones. So the parietal cells then are going to produce hydrochloric acid. And the main function of hydrochloric acid is to provide the acidic pH, to denature protein, to cleave pepsinogen into pepsin, to break down any kind of cellulose, and to kill as many bacteria as possible. And so here in the stomach, we have the first line of defense in our immunological system, and that is a chemical barrier in the form of hydrochloric acid. So look at this. I'll give you a few seconds here to write down, or you can pause if you'd like. All right. How do we remember that pepsinogen and pepsin break down proteins? Pep for peptide, and peptides make up proteins. So pepsin breaks down proteins. The parietal cells also produce intrinsic factor. Intrinsic factor is incredibly important to the well-being of our body. It is necessary to pick up vitamin B12 in the small intestine. It picks it up in the stomach and in the small intestine, but in the small intestine, an intrinsic factor facilitates the absorption of vitamin B12 into the circulatory system. And this keeps us alive. This is important. It keeps us alive. The chief cells are going to secrete two enzymes, pepsinogen, which is then going to be activated by HCl and become pepsin, and then lipases. Remember, lipases are enzymes that break down lipids. Most of lipid digestion will take place in the small intestine, but some of it begins to take place in the stomach. Enteroendocrine cells. Let's look at that word. Entero means in the intestines or in the gut. So consider the stomach part of the gut. So entero, digestive, in the gut. Endocrine means, of course, the endocrine system, hormones. Remember that we have hormones that are produced locally and act on the tissue that surrounds it and close by tissues. And these are called paracrines. Paracrines. If you need to review, go back to the endocrine system chapter and look at those definitions. So the two paracrines are serotonin and histamine. We'll talk about their functions in a few minutes. The hormones, somatostatin, which can act as a paracrine in this case, and then the other hormone is gastrin. If you would like to think of these four as hormones, that's fine with me because that's what I do. I just remember that they work locally, and that's why they're called paracrines. So serotonin, histamine, somatostatin, and gastrin. So this picture is very good at showing you the gastric gland and telling you that, okay, here are the parietal cells, secretes HCl, an intrinsic factor. The chief cells secrete pepsinogen, and it's the hydrochloric acid that cleaves pepsinogen into pepsin, and then pepsin breaks down proteins in the stomach. And then we have the enteroendocrine cells. These are the green ones, and they secrete hormones. They secrete hormones, the four hormones. Since so much hydrochloric acid is produced by the stomach lining has to be protected. And the barrier is made up of mucus and bicarbonate. So what's bicarbonate plus HCl? That's going to be very important to maintain the proper pH and to make sure that the stomach lining is not destroyed by the hydrochloric acid. We keep God prepared tight junctions between the epithelial cells so that the gastric juices don't seep in between and destroy underlying tissue. And what just blows my mind is about once a week, the entire surface of your stomach is replaced. The entire surface. That's fascinating. But that keeps our stomach tissues safe. What are some of the diseases that we see? Well, gastritis, so inflammation of the stomach's mucosal barrier. A lot of times this is an upset stomach that can lead to vomiting, even diarrhea, depends on what the culprit may be. And then we have peptic or gastric ulcers. These are ulcers found in the stomach proper. There's another set of ulcers called duodenal ulcers, and they're usually found very close to the pyloric sphincter in the duodenum. Most of the gastric ulcers are caused by a bacterium called Helicobacter pylori, and it can ulcers can also be caused by non-steroidal anti-inflammatory drugs, NSAIDs, that would be aspirin, ibuprofen. So that's why we need to tell our patients not to take too much aspirin or ibuprofen. Don't take beyond the prescribed dosage. Here's a picture of a gastric ulcer. You can see the hole in the mucosa and the dead or dying tissue underneath at the bottom, and you can see how the tissue around it is highly inflamed. It looks very angry. It's all red and nasty and highly inflamed. This is very, very painful. Very painful. And here's a colored micrograph of the H. pylori sitting on the surface of the stomach. They secrete enzymes that break down the surface of the stomach so that they can have something to chow down on. Notice they have flagella so that they are definitely motile. This might be a nice time to take a little break, put it on pause before we go into the digestive processes of the stomach. How does digestion occur in the stomach? Well, we have to remember that we have mechanical digestion, we have chemical digestion, and then also it stores our food. So, not everything that we eat goes directly into the duodenum. It takes time to make a chyme so that the brush border enzymes in the small intestine can have better access to the nutrients, and then it's easier for those nutrients to be absorbed into the hepatic portal system. If they're in their monomers. All right. Notice that in infants, renin breaks down the milk protein, casein, and that's why it looks so chunky. Looks like cottage cheese because we use cow renin to break down the casein in milk to make cottage cheese. All right. Anything that is lipid soluble, as well as the NSAIDs, are going to be absorbed into the blood in the stomach. And a fascinating fact, this is great to wow and impress your friends and family, is that really the only absolutely essential function of life the stomach has is to produce intrinsic factor so that B12 can be absorbed. Yeah, this is why a lot of times with people who have gastric bypass surgery initially have anemia, or they may have anemia the rest of their lives since B12 is necessary for the red blood cells to mature, and since you've cut off access to 2/3 of their stomach, there's not enough intrinsic factor being made and absorbed so that B12 can't be picked up and absorbed. So we have to give them B12 shots that can be absorbed directly into the musculature and then into the capillaries in the musculature so that they don't have anemia. This kind of anemia is called pernicious anemia, and it can be deadly. Another fascinating fact is you produce a little more than 3 liters of gastric juice a day. So that would be about 3 quarts of gastric juice a day. Now this secretion is going to be regulated by the nervous system and the hormones. So remember that the parasympathetic nervous system increases secretion, and the sympathetic nervous system is going to decrease secretion. What do the hormones do? Well, gastrin stimulates hydrochloric acid secretion, and then it can be used as an antagonist hormones by the small intestine. Okay? So let's look at this. The regulation of secretions. There are 3 phases. The cephalic, which is a reflex. Cephalic, think brain, think nervous system. The gastric phase and the intestinal phase. So the reflex phase, you smell or see, taste, even think about food, and your brain says, that sounds good, or that smells good. I don't know about you, but frying chicken always makes me hungry. So I smell Chick-fil-A on campus, and my brain says, ooh, fried chicken, and sends a thought to the brain, sends a signal to the stomach, and the stomach says, yeah, that sounds good, feed me. The gastric phase can last 3 or 4 hours, and during this time, most of the gastric juice will be released. What stimulates this is what we call the long and the short reflexes. If you eat something, it's going to distend your stomach, fill your stomach. Those stretch receptors will react and say, time to go to work. And then the chemical stimulus, food, nutrients in your stomach, caffeine, low acidity, these are going to activate the hormone G cells, we'll just say the hormone cells, to secrete gastrin. During the gastric phase, we're going to see that gastrin is going to cause hydrochloric acid to be released from the parietal cells, and then HCl is going to break down pepsinogen into pepsin, and pepsin is going to break down polypeptides into smaller peptides. It also stimulates the release of histamine, all right? Stimulates the release of histamine, and then we're going to have the buffering action of the proteins. Remember, proteins are not as acidic as HCl, and so it's going to buffer the HCl, and that's going to activate even more gastrin secretion. So the sympathetic nervous system then is going to inhibit. So low pH, in other words, if your stomach is empty, you're going to have a lower pH, and that's going to inhibit gastrin from being excreted. And if you inhibit gastrin, then hydrochloric acid won't be produced. So the more protein we have, then the more HCl is secreted. Okay? the more HCl is secreted, and then that has a decline in pH. So this is going to be toward the end of emptying the stomach, where we see a lot of HCl has been produced in response to high protein. That makes the pH go down, and that inhibits the gastrin. The intestinal phase. So basically, when the chyme is propelled into the small intestine, into the duodenum, then that is going to stimulate the duodenum to release intestinal gastrin. Intestinal gastrin. It's also called enteric because it's in the intestines, enteric. It's easier to remember gastric gastrin or stomach gastrin, and then intestinal gastrin. And so what intestinal gastrin does is cause the gastric glands to continue their secretion, and then once the small intestine, really the duodenum, gets pretty full, then intestinal gastrin will decrease so that the digestive juices will be inhibited. Isn't that cool the way God made that? So what is going to inhibit the intestinal phase? 1, distension of the duodenum. 2, the presence of acidic chyme coming in from the stomach. The presence of fatty chyme. Fatty, remember, only about 15% of lipids are going to be digested in the stomach. The vast amount of lipid digestion takes place in the small intestine. And then the presence of hypertonic chyme. Hypertonic. So these 4 factors in the duodenum are going to cause the inhibition of the stomach to produce the gastric juices. Okay. So the enterogastric reflex, the duodenum inhibits the production of hydrochloric acid by what's called a short-term reflex. And that means it goes from the intestine to the stomach. It can also stimulate the sympathetic nervous system and the vagus nerve, the parasympathetic nervous system, and the long reflexes. Enterogastrones are hormones that the duodenum is going to secrete in order to inhibit the stomach from making more gastric juice. And these are called secretin and CCK. Secretin and CCK. Notice that CCK starts with cholecystokinin, and we remember that chole is going to refer to bile, and kinin is going to be a protein that is a hormone. All right, so take time, pause, and really study figure 2319 so that you understand the cephalic phase, the gastric phase, and the intestinal phase, what stimulates each phase and what inhibits each phase. This is an excellent little graph or chart, and it summarizes everything I've just talked about. This chart has lots of good test questions on it. So pause here, or maybe even print this off if you want, and make notes and study this. Here is a table about hormones and digestion. And so I want you to know the following hormones. You have your pencils ready or your pens. I want you to know the hormone and the activity. The hormone and the activity. And as you learn about the hormone, you're going to remember where it's produced, what it does, and where it goes, the target organ. The hormone and its activity. So the first hormone is CCK. CCK. The second hormone is gastrin. Gastrin. This is stomach gastrin. All right. The second hormone is histamine. Histamine. Quick note, histamine activates the release of HCl, and if you take a lot of antihistamines to control your allergies, you're also inhibiting the parietal cells from releasing HCl. That's why sometimes physicians will give an antihistamine to people who are having problems with ulcers to help lower the secretion rate of HCl. The next hormone is intestinal gastrin. Intestinal gastrin. So, CCK, check. Gastrin, check. Histamine, check. Intestinal gastrin, check. Now on this next, or the second half of this chart, notice that motilin stimulates motility. You should look at that and say motilin stimulates motility. I know it says stimulates migrating motor complex. Yeah, motility. So the next hormone that I want you to know is secretin. Secretin. Serotonin. Serotonin. And then last, somatostatin. Somatostatin. Secretin, serotonin, and somatostatin. Know the hormone and its activity. And when you learn its activity, you will learn its site of production and its target organ. So let me go over that list one more time. You ready? CCK, gastrin, histamine, intestinal gastrin, secretin, serotonin, somatostatin. You probably remember serotonin as a hormone that also works in the brain, and you're right. All right, why don't you take a little break, stretch, come back in a couple, 3 minutes, and we'll talk about how hydrochloric acid is formed. Hydrochloric acid formation is very important. And if you remember from our studies in gas exchange, you remember that carbonic acid is formed and absorbed by the circulatory system as a way of transporting CO2 out of the tissues and into the lungs. So carbonic acid can be absorbed from the capillary system, okay, and it's broken down into bicarbonate and protons. Remember that this is done by carbonic anhydrase. So the parietal cells in the gastric gland are going to pump protons into the stomach, and this is going to be an active transport. It's called the proton potassium ATP pump, ATPase, also known as proton pumps. You may have heard of drugs called proton pump inhibitors, and these are used to decrease the production of hydrochloric acid in people who have acid reflux, to try to help control the amount of HCl that the stomach produces. So once that protons are pumped into the stomach, the bicarbonate ions are exported back into the blood via the chloride and bicarbonate anti-transporter. And we talked about these different kinds of ways to transport ions way back in the fall. And so basically what happens is bicarbonate is exported into the blood, okay, and chloride is transported into the stomach. Bicarbonate is exported into the blood, and bicarbonate is transferred into the stomach, so that when chloride is pumped into the lumen, it joins with the protons to form hydrochloric acid. The fact that the bicarbonate is pumped out of the stomach and into the blood is called the alkaline tide. The alkaline tide. And all that means is bicarbonate is leaving the stomach and going into the blood. Excuse me. So this picture gives you the steps of how HCl is secreted. So take a moment, pause, and go through this picture so that you understand what's taking place. These 4 steps. If you have questions, please email me and I will be happy to visit with you about how this takes place. So here we go with mechanical digestion. We've talked about the chemical digestion and the enzymes, the hormones. Now let's look at the mechanical digestion. So in the stomach, there are peristaltic waves and segmentation waves. And what we see is that the peristaltic waves move from the top of the stomach, the fundus, to the pyloric area, the pylorus. And that makes good sense because you want the chyme to go into the small intestine. Very important. There's grinding. Most vigorous peristalsis and mixing is going to take place in the pylorus. Okay? And then the pyloric area is going to pump chyme into the duodenum through the pyloric valve. Sometimes it's called the duodenal pyloric valve. Sometimes it's called the duodenal valve. Mainly I've heard pyloric valve. So here we have, and notice that chyme is going to come out in small squirts. This is so the acidic nature can be fully neutralized by the pancreatic juices. You don't want fast stomach emptying because what that means is that nutrients are not broken down properly and they're not absorbed properly because everything moves through the stomach and the intestines too fast. And then we have retropulsion. And what that means is that the pyloric valve closes and then the contents of the pylorus are going to move backwards into the stomach. So you have basically up, down, up, down, up, down, up, down. And then at the same time, you can have the stomach muscles contracting in the other two dimensions. And this is mechanical digestion. So what regulates the stomach emptying? Mainly the duodenum. You have the reflexes, the enterogastric reflex we've already talked about, and the enterogastro hormones, which we've already talked about. So the stomach is going to empty in about 4 hours. Some people a little bit longer, other people a little bit shorter. But if we eat a lot of high protein and fat, let's say you go and have tamales, and that is going to make your tummy full for up to 6 hours or more because of the protein and fat concentration. This is why high protein meals and fatty meals make us feel full or satiated. We're happy, we're full for longer than, say, a high carbohydrate meal. So any carbohydrate breaks down fast and moves through the stomach and the duodenum very quickly. And so if you go and you eat fried rice and you don't have much protein with it, you're hungry again in a couple of hours because your stomach's empty, because carbohydrates go through the stomach really fast. The moral of the story is eat more protein. All right, draw a line in your notes and write down liver. Going to talk about the liver now. You learned about the anatomy of the liver. What I want to point out to you here is the hepatic portal vein. So everything that is absorbed into the bloodstream in the intestine is going to be dumped into the hepatic portal vessels and transported to the liver so that all those nutrients can be used by the liver and sent out to make proteins and carbs and fatty acids and all the other stuff that we need, and also transported out into the rest of the system via the heart. Also see the hepatic artery proper, and then the common hepatic duct. The common hepatic duct is where bile is going to be transported from the liver to the hepatic duct. In the hepatic duct, the cystic duct splits off of it so that bile can then be stored in the gallbladder. What's not stored in the gallbladder goes directly into the duodenum. Let's look at the microscopic anatomy for review. Remember that the liver cells are called hepatocytes. Hepato means liver, cyte means cell. And that these hepatocytes are responsible for filtering the blood and processing nutrients into polymers. Really, really important. In the middle of the liver lobule, you see the central vein. And then, very important are the portal triads at the corner of each lobule. The portal triads. You will need to know what vessels are in the portal triads. We have the hepatic artery that supplies oxygen. We have the hepatic portal vein that brings the blood from the intestines to the liver. And then we have the bile duct. And basically, it is collecting bile that's being made by the hepatocytes and shunting it to the duodenum and the gallbladder. So these are what liver lobules look like. Very nice picture. So a lobule is a functional unit of hepatocytes, and notice in the center you have the central vein. So here's it up close and personal. What are the liver cells called? That's right, hepatocytes. Okay, here is a schematic, a drawing, showing the portal triad. Now in your body they're not really red, blue, and green, but that's color coding that humans use. So remember that the bile duct receives bile from the liver. Okay, we have fenestrated capillaries. Remember? Sinusoids, fenestrated capillaries. Go back to the cardiovascular chapter and review capillaries. Artery is going to deliver oxygen-rich blood, and the hepatic portal vein is going to deliver the nutrient-rich blood from the intestines. The central vein then collects blood from the lobule. It's going to take it to the hepatic vein. The hepatic vein is going to join the inferior vena cava, and the inferior vena cava is then going to dump into the right atrium of the heart, go through the lungs, go back through the heart, be pumped out by the left ventricle so that the rest of the systemic tissues can receive nutrients. All right. Liver sinusoids, remember these are the capillaries. Very important to our immune system are the hepatic macrophages. And also the liver contains natural killer cells and neutrophils to help control and kill off bacterial infections. Your body produces about 900 milliliters of bile per day, processes all the nutrients. It can store glucose as glycogen. It can make plasma membranes. It can make bilirubin. It can detoxify the ammonia that is a waste product from tissue metabolism into urea. It detoxifies alcohol into formaldehyde, which is why we have problems. It stores the fat-soluble vitamins. It's just a phenomenal organ. It makes bile, and bile is kind of yellow-green looking. It's alkaline, so it's going to have a high pH. And it also makes the bile salts. These are derived from cholesterol, and these salts are necessary to emulsify fat, so it can be broken down by the intestinal lipases, and then the fatty acids can then be absorbed by the intestine. It also forms bilirubin, which bacteria in the gut will break that down, the bilirubin, into stercobilin, and that gives our poop its brown color. So if your poop ever goes white and you haven't had any kind of test done, that means your gallbladder is either clogged with gallstones or your liver's not producing bile. Yeah, don't want that. Don't want that. So this is a lovely picture of the intrahepatic circulation, or what I call the hepatic portal system. Please pause here and read through this picture and make sure you understand the 4 steps of the hepatic portal system. What else does the liver do? Well, how about what if it doesn't work? Hepatocitis, which is inflammation of the liver, and this is usually due to a viral infection. Could be drug toxicity. Lots of different poisons can cause this problem. So hepatitis A, B, C, D, E, F, G, H, but mainly we see A, B, and C. Cirrhosis of the liver is a chronic inflammation from chronic hepatitis, and chronic hepatitis can be caused by a viral infection or by drug abuse or by alcohol abuse. Basically what happens is that the hepatocytes are replaced by fat cells and scar tissue. This causes portal hypertension, and so high blood pressure in the liver, and that can cause all kinds of major problems down the road. People can get liver transplants, but it is hard to find livers. What fascinates me about the liver, and I think God did something awesome here, is that if you remove 75 to 80% of the liver, it can regrow, it can regenerate, not scar tissue, hepatocytes. It can regenerate back to its full size within a year. So they can take one liver from a donor and split it between at least 2 people, sometimes 3 if one's a child, and 6 months to a year later, they've got a whole liver that functions. That is phenomenal. That's amazing. Way to go, God. God also gave us this little green pack, and it really is green on the inside. It's called the gallbladder. And basically it's just a sac, stores bile, and it concentrates the bile. Okay. It is going to release bile into the duodenum through the cystic duct. The cystic duct will flow into the bile duct, and then the bile duct will dump into the duodenum. Major problem, and if we were in the classroom, I would bring out my bucket of gallstones and show you all the different kinds of gallstones that have been removed by my friends who are surgeons, and they give me samples so that I can show you guys. So when we get back to the classroom, please remind me, and I will bring out my bucket of gallstones. But basically what happens is that the diet's too high in cholesterol, or the liver's not making enough bile salts, and oftentimes it's both. And these stones just start forming in the gallbladder, and they can be as large as a nickel. They can look like sand, but it obstructs the gallbladder, and it's very painful because when the gallbladder contracts, and it contracts against these stones, and that's when people feel like, oh, I think I'm having a heart attack. If the blockage causes bile salts and pigments to build up in the blood, then people get jaundice. but people usually are jaundiced because of the fact that the liver is failing. It's not breaking down the bilirubin. How do we treat gallstones? Well, first they can try drugs, and then what we call lithotripsy, which is basically they shoot sound waves into your gallbladder and try to break up the stones into itty-bitty pieces so that they will flow out. They can use lasers to zap them, and most of the time you can do surgery. Usually when someone has to have surgery, everything else has failed. A lot of times people don't know they have gallstones until it becomes a crisis and they need emergency surgery. Wait, draw a line and write the pancreas. The pancreas. When we look here at the pancreas, remember that it hugs the greater curvature of the stomach. The head is going to be connected to the duodenum, and the tail is going to be over by the spleen. It has endocrine functions in that it produces insulin and glucagon by the pancreatic islet cells, and we've already studied these when we studied endocrines. And then it has exocrine functions. So exocrine is basically the production of the pancreatic juice. It's done by the acinized cells, and they produce zymogen. These granules contain proenzymes. Sometimes people call them pre-enzymes. So pepsinogen is a proenzyme to pepsin. Let me say that again. Pepsinogen is a proenzyme to pepsin because pepsinogen is secreted. It is the inactive form. Hydrochloric acid cleaves pepsinogen to release the active enzyme pepsin. Pepsin breaks down proteins. And then the ducts of the pancreas are going to produce water and bicarbonate by the duct cells, which are going to join the proenzymes and then be pumped out into the duodenum. So here's a picture showing the acinar cells. It's called an acinus because it's kind of round. Okay. You see the zymogen granules that contain all the proenzymes. And then the gland cells, or the duct cells secrete bicarbonate and water. Bicarbonate and water. Where's the bicarbonate come from? Do you know? You're right. It comes from the breakdown of carbonic acid by carbonic anhydrase into protons and bicarbonate. Very good. Comes from the blood. Where does the water come from? Yeah, it comes from the blood. Here's a micrograph of the acinar cells. So you produce 1.2 to 1.5 liters of pancreatic juice a day that consists of an alkaline solution. It's at a pH of 8, and it's going to join with that lovely chyme that has a pH of about 3, and it neutralizes the chyme. This is good because the intestinal enzymes need to have a pH of about 6 to 8 in order to work properly. It also contains electrolytes, proteases, which break down proteins, okay, amylases to break down carbohydrates, lipases to break down lipids, and nucleases to break down the nucleic acid. Remember that enzymes are going to break down polymers into monomers. In the digestive system, enzymes break down polymers into monomers. Those monomers are then absorbed into the hepatic portal system, sent to the liver and the rest of the body. The bile duct and the pancreatic duct fuse to form the hepatopancreatic ampulla. Can you just remember that the bile duct and the pancreatic duct dump into the duodenum? Okay, dump into the duodenum. So here we have a picture. Locate the duodenum. See the major duodenal papilla, which is where the pancreas and the pancreatic duct and the bile duct dump the pancreatic juices with the enzymes and the bile into the duodenum. And you can see that it's right there by the pyloric sphincter, so it can neutralize the acidic chyme. Note the position of the tail and the head of the pancreas and the position of the gallbladder. The hormones that are necessary to control bile and pancreatic secretions are CCK and secretin. What's going to increase bile is when there's the return of bile salts. Secretin is exposed to HCl and phytic chyme. Will stimulate the gallbladder to release bile. And then when the hepatopancreatic sphincter is closed, the bile is stored in the gallbladder. This ends lecture 2. I know it's long, but please take the time to go back, listen to it 2 or 3 times, check your notes. Please make sure you do your learning objectives. Remember that your test questions will come from the material covered by the learning objectives. So make sure that you're doing your learning objectives. I will post this lecture 3 and 4 in the near future.